Looking good.
Great. Thank you, David. Thank you for the invitation today. A disclaimer at the outset, I am not a geneticist, so many of the things that you have been talking about today are slightly foreign to me, though David and Rob are doing their best to educate us. I'm going to talk today about the connectome, which I would consider a very complex trait, though several of the talks I've just seen humble me. There are many more complex traits. I'm spending a little bit of time telling you what this connectome is. It is a method that is derived from MRI. And in MRI, if we look at this cartoon and measure the diffusivity, we can look at the mobility of water, diffusion of water. And in a cell, in a neuron, that diffusion is isotropic. That is, it can move in any direction without constraint. But along the projections, the axonal projections, the diffusion is not isotropic. It is constrained by the myelin bundles and the structure, the cytoarchitecture. And this can be measured in MR by applying a magnetic field gradient during the acquisition. As the arrow tends to, this is the image on the left is an image of a mouse brain. This is a perfusion fixed mouse brain, what we call magnetic resonance histology. And in the center, we have applied a gradient that moves in the direction of the arrow. I draw your attention to the anterior commissure here. You see it's a little darker here, a little bit lighter here. If we reverse this gradient and put it in another direction, we see the opposite effect. And so the signal is decaying as the water protons diffuse along the direction of the gradient. And we can exploit that to map out the circuits of the brain. And a few years ago, with Rob and colleagues at UTHSC, Nian Wang here in the lab developed a method to look at the connectome across 4 strains. We hypothesized that the connectome, like a facial feature or like any phenotype, would be highly heritable. And we started with just 4 strains of animals: the B6, the D2, the CAST-J, and the BTRB. And we included the BTBR because it is a known phenotype for autism. And what you're seeing here are tractography. That is, we use this projection method to map out the tracks in the brain. On the left, you see the tracks for the C57. On the right, the BTBR. The color, the green, suggests movement from anterior to posterior. The red is showing us tracks that are moving across the brain, left to right, and blue prone in and out. And so look here at motor cortex. We see the motor cortex is projections into the motor cortex are considerably altered here in the BTBR. We see in the cerebellum there's a lot of disruption. We see just a sort of a messy mess of projections here. So this can be quantitated if you can segment the brain into a series of regions and map the strength of connections between those. And that has been done. We're using the common coordinate framework from the Allan Brain Atlas, which we have mapped into a pipeline that lets us label all of the brains. So we scan a bunch of brains, we label them, and then we generate the connectivity from region to region. So this schematic here shows all of the regions that we've outlined and all of the regions that we've outlined. So if we look at the hindbrain here, and we go to the hindbrain and say, how connected is this region of the hindbrain to isocortex or to pallium or to subpallium? The color is a log 10 scale. So there's enormous dynamic range in this. And each entry here is one region of interests, the strength of connectivity between that region and the corresponding region along the axis above. And you can see that we go over about 8 or 9 orders of magnitude. The red are the very highly connected. Each dot, each entry here is an edge. The strength of that edge then tells us how well connected that is. If you look across the strains, I've blown up the connectome for the B6 and the BTBR here, and I've taken this one line here that's labeled and blown it up here. This is a region in the corpus callosum. And we're looking at that region in the corpus callosum and the strength of its connectivity to everything else in the brain. And the red and yellow are highly connected. The blue are less connected. This is the left half of the brain and the right half of the brain. And it becomes sort of an across the room map in the sense that you see that the connectivity to the contralateral side, from the left to the right, is reduced. This is a known phenotype of the BTBR. So this was encouraging to us. We included the BTBR because we knew we were pretty confident that we would see some difference. Where we were less confident was whether we would see differences just within the more common strains. And we could compare those with a fairly sophisticated statistical method in which we go to a node, a region, this region of the hippocampus or of the corpus callosum, for example, and we express everything else out here as a vector describing that region. And then with some complex math that is frankly just barely conceivable by me, our colleagues at Johns Hopkins developed this method to reduce this sort of like a principal component analysis. And when we plot that, we see a wonderful clustering between, here's the BTBR clustering far from the others, but we also see differentials between all of the quote, quote, common strains or very similar strains. And when we take the BTBR out of the picture altogether, we see separation that is much even more dramatic. So this gave us glorious opportunity to start saying, well, hey, this connectome can be very useful in what we look at. Can we identify specific regions of interest? Can we look at a given node, for example, that corpus callosum or the substantia nigra internal capsule? And statistically, these all turn out to be highly heritable. And you can see that in the tractography, where we are seeding just the internal capsule or just the substantia nigra, where we're casting computer seeds into those regions of interest and say, show us where the projections go. Visually, it is quite stunning how well they are differentiated. So we have begun moving this out into a study of aging, and we've asked the question, well, is there a strain dependence on the change of the connectome with age? I am living proof that the connectivity goes down with age. I've lost a lot of it. The only thing left is my humor centers, but they seem to still be working. And we're asking, is the change heritable? So this is some work which Catherine Hornberg has done with David and Rob, in which we've looked at about 114 different animals, 13 BXD strains, with a very high spatial resolution. This is about 90,000 times the spatial resolution of a clinical DTI scan. These are all perfusion fixed, that is post-mortem studies, and then quantitative connectomes generated. We have two clusters of ages down here around 100, and then up here at around 420, 430 days. And these are the embedding directions for the entire connectome, where the old are purple and the young are green. This is all of the strains together. When we begin to sequester just a given strain, we look at the BXD65 and we see the separation. First of all, we see clustering that's a little bit tighter for the younger animals. It disperses with age and separates. This unit, the embedding dimension, is defined by the range of studies that you're looking at. So the BXD65 was mapped at the same time as the BXD34, and you see that the BXD34 does not separate much at all, but the BXD32 does. We can map this across all of the strains that we looked at, and the BXD65 we're really mapping right now. This is the number that we're mapping, the separation for this stage. And in this, we see that this is highly dependent upon strain. BXD65 is very vulnerable to aging. The BXD34 is really quite resilient. So this gives us confidence that we can look at the connectome as a measure of aging. I would be delighted to be looking at the connectome. We have previously looked at something less sophisticated with colleagues at UNC looking at fetal alcohol syndrome, and we see changes there. We are hoping to be doing some work with Megan Mulligan looking at the impact of cannabis on the connectome. We think this is a very powerful phenotype that can tell us quite a bit about the brain and the resilience of the brain to challenges. With that, I will close and be happy to entertain questions. I'll leave you with a little eye candy. This is a tractography image done at 5 microns through the whole brain, where the color is indicating the direction of the projection, and the intensity is how strong is the connectivity there. There's more data that we can digest here. There's about 2 billion pixels in this image, and I hope that blows your mind just a little bit. Happy to answer questions.